So with this, let us see how various body fluid compartments are measured. For that, first we should know what are the characteristics of the substances which can be used to measure the body fluid compartment. First thing, that the substance should be non-toxic, it should be non-harmful to the body, isn't it? Then it should be easily measured. As I told you that we have to take out a sample and measure its concentration. So it should be easily measured. Then it should be excreted and metabolized very slowly, because if it is very fast metabolized, then the mass will change tremendously, isn't it? And the mass may change so much that there might be hardly any concentration in the body. So it should be metabolized very slowly. Then it should distribute into concerned compartments only. That means if we want to measure only the plasma, then the substance should go only into the plasma. If we want to measure say suppose entire extracellular fluid, then it should distribute both in the plasma as well as in the interstitial space. Okay. And finally, it should distribute evenly across compartments, as we saw in the previous picture that how evenly it is distributed. This is important because when we take out a sample from one particular region, that concentration should be representative of the concentration in the entire compartment. It should not be that at one place it is more and at other place it is less. Okay. So it should distribute evenly across compartments. So with this concept, let's try to understand which substance we can use to measure various body fluid compartments. See, remember, whichever compartment we have to measure, the substance should go into that compartment and not more than that. Okay. So these are the compartments which we had seen. Now see, water can cross all compartments freely. Okay. Then ions. Ion can cross the capillary membrane. So they can be in the blood vessel and they can cross the capillary membrane and they can be there in the interstitial space. Then proteins. Proteins cannot cross the capillary membrane. Okay, so they will remain only in the blood vessel. Why are we talking from the point of blood vessels? Because whatever substance we use, we have to inject into the blood vessel. So from blood vessel, if we inject, water can cross anywhere, ions can cross into the interstitial space and back and forth, but not into the intracellular fluid, and proteins cannot cross the capillary membrane. So they will remain in the blood vessels only. So let us see which are these substances. So which substance can be used for the measurement of total body water? Basically, we want a substance which goes freely into various compartments. So we have said that water which can cross all the compartments. So if it is injected, it can cross all the compartments, that is it will be in the blood vessels, in the interstitial space as well as in the intracellular fluid. So that's why we use radioactive water, that is tritium, then heavy water, that is deuterium, and there will be another substance, that is antipyrine, which can also cross the various compartments and can be used. For the measurement of extracellular fluid, that is the interstitial space and the plasma, which substance can be used? Yes, we said ions can cross across this capillary membrane and they can go into interstitial space and back and forth into the blood vessel. So yes, we use radioactive sodium, which is the ion, and we said that it is the major cation also of extracellular fluid. So we use radioactive sodium. Why we are using radioactive? Because we have to tag whatever we are injecting, we have to tag it, right? Because we want to measure it later. Okay, so radioactive sodium is used. Then there is radioactive chloride which we can use. Then another ion, that is radioactive iodothioamate, can be used. Then thiosulfate ion can also be used. And there is another substance, inulin also, which can be used. Now, it has been found that when we use various substances to measure extracellular fluid compartment, there are some minor differences between that measurement. So instead of using the term that this is the value of extracellular fluid compartment, we use the terms like sodium space and inulin space depending on which substance we are using. Okay, so it will give us an idea that why this difference in extracellular fluid concentration is there. Okay, so instead of ECF we use the term sodium space and inulin space. So which substance should be used to measure plasma volume? Well, it should remain in the blood vessels and should not cross the capillary membrane. So we have seen that proteins do not cross the capillary membrane. So we use a protein, that is albumin, which is tagged with the radioactive iodine. And there is another protein, that is the Evans blue dye, which is used for plasma measurement. So with this we have determined three compartments, that is the total body water, extracellular fluid and the plasma volume. But what about the intracellular fluid, that is the fluid present in the cells? If we want to measure this intracellular fluid, we want that we should inject a substance, it should cross the capillary membrane, it should cross the cell membrane, it should go inside the cells and stay there. It should not come back into the blood vessels because we want that the substance should distribute only in that particular compartment. But this is not possible. So ICF cannot be determined directly. However, you see that we can determine total body water by using radioactive water, and we can determine extracellular fluid also by using our radioactive ions. So if we determine that and subtract extracellular fluid from total body water, we will get our intracellular fluid. Similarly, we have not determined interstitial fluid because there is no substance which will go into the interstitial fluid and remain there. Okay, so this also cannot be determined directly, but we can determine ECF and we can determine plasma volume. So if we subtract the determined plasma volume from the determined extracellular fluid, we will get the interstitial fluid. Okay, but you remember that apart from these, we also spoke about blood volume, right? So we need to determine blood volume also. How it can be determined? It can be determined using plasma volume. See, if you remember, we said blood volume is basically 60% plasma and 40% RBCs. So if we determine the plasma and we know the hematocrit, that how much percentage is the RBCs. So this is just a general value, 60% plasma and 40% RBCs, right? But it can vary from person to person. So we need to determine something known as hematocrit. Okay, that is the percentage of the blood volume occupied by RBCs, right? If we know that, we can use this particular formula, that is blood volume is equal to plasma volume divided by 1 minus hematocrit, and we will get the blood volume if we know the plasma volume and the hematocrit. Or there is an alternative method. We can inject radioactive chromium labeled red blood cells into the blood volume and we can determine the concentration of these RBCs. Okay. So that's how we determine various body fluid compartments. So that's all about body fluids, their ionic composition and how they are measured. Thank you. Have a nice day.